Hello everyone. Today we're going to discuss different types of microscopes that we use to observe microorganisms, and we're also going to introduce the various staining methods and stains that we use when we are trying to observe those microorganisms. Light microscopy. So light microscopy, those are basically microscopes that use visible light to allow us to observe those microbes or specimens in general. Okay. Now it turns out that there are so many different microscopes out there, and they work differently. We have the compound light microscope. The compound light microscope is the most common light microscope that are used. So in our microbiology lab, we use compound light microscope, and you're going to notice actually that the compound light microscope, it is the microscope used in university and college settings. This is the microscope that we use, compound light microscope. But there are different types of light microscopes out there. There's the dark field, there's the phase contrast, there's the DIC, fluorescence, the confocal microscopes. There's so many different types of light microscopes out there. This is how a compound light microscope looks like. So this, let me kind of show you the parts. So right here, basically this is the base, okay? So you set your microscope on your countertop, the base is down here. Right here, actually, there is a little kind of knob where you can control the lighting, okay, how much light you want to shine on your specimen, if you want to make it too bright or if you want to dim the light, okay, so this is an option for you to dim the light. You have those knobs right here. There's the coarse knob, which is going to help you, by rotating it, you can go ahead and start viewing the specimen. If you want to focus your image because it's blurry, you can go ahead and use the fine focus knob. By the way, the light is going to shine from down here, so you have like the light source from down here. So the light is going to shine, okay, from the light source, and it's going to shine upwards on the specimen. So where do we place our specimen? We place our specimen right here on the stage. So on the stage, you go ahead and you place your slide that has the specimen. Down here below the stage, you have the condenser, which is going to focus the light onto your specimen. And then there is the iris diaphragm, or you can just refer to it as the diaphragm. And this one controls the amount of light. Okay, so this one controls how much light do you want to, again, shine on your specimen. And then the condenser focuses it on your specimen. So as you can see right here, so the light is going to come out from the illuminator. You can, as I said, control how much light and focus it on your specimen, and it's going to hit your specimen. It's going to then hit the objective lenses, okay? And these objective lenses, we have a bunch of them, as you can see, and each objective lens can magnify your specimen differently. So one can magnify your specimen 4 times, 10 times, 40 times. In microbiology labs, we usually use the objective lens that can magnify our image 100 times. Okay, we call it the oil immersion lens, and I'll show it to you. And so the light is going to hit the objective lenses, and then it's then going to move up, and it's going to also pass through the ocular lens. And by the way, the ocular lens also magnifies your image. So if you think about it, you're actually magnifying your image or your specimen twice. The ocular lens is going to magnify it, and the objective lens is going to magnify it. So at the end of the day, we say we have to figure out the total magnification. To figure out the total magnification, you multiply the power of your ocular by the objective lens. Let me give you an example. So let's say you are going to use the oil immersion lens, which is 100x. So meaning it can magnify your image up to 100 times. And your ocular is magnifying your image 10 times. So this one is magnifying my ocular, magnifying my image 10 times. The oil objective is magnifying it 100 times. What do? You multiply those two numbers. So 100 multiplied by 10, that means I am magnifying my specimen by a total of 1,000 times. Okay, so multiply the two to figure out your total magnification. There are a few terms that you need to know. So there is a term known as the resolution or the resolving power of a microscope. What's a resolving power of a microscope? It's basically the ability of your microscope or the ability of the lenses, right, of your microscope to distinguish two separate points. Meaning, if this is what you're seeing under the microscope, this white, kind of blurry, fuzzy circle, okay? Right here, how many white circles are you viewing? Just one, okay? But actually, you're seeing it as one because this is a microscope with a pretty bad, pretty low resolving power. If you go ahead and use a microscope that has a better resolving power, so you guys see how you're starting to see the two separate circles, okay? And then right here, that's even better. This microscope, it's actually right, this top one is actually the best because with this microscope, you are actually able to see that you have two separate circles. So do you see the big difference between using a microscope with a low resolving power? It's going to show it to you as if as if that circle is just one, versus using a microscope that has a pretty good resolving power. You can actually see the two separate circles. So that's what the resolving power is all about. There's another term called the refractive index, and refraction of light is basically the bending of light. So light has the ability to bend, and light usually bends when it hits two different media, meaning right now there is light hitting your eyes, right? And that's why you're able to see, because the light hits your eyes, etc. So the light hitting your eyes, the light shining into your eye, this light is actually, if you think of the light beam, it's actually traveling in air first, right, before it hits your eye. So it's traveling in an air medium, okay? And then when it hits your eye and goes inside of your eye, there is liquid medium. So the condition inside of your eye, there's liquid in there. And so what happens when light goes from air into liquid? The light is actually going to bend. And we refer to it as refraction. So it bends inside of your eye. And that's what also happens, when we place a specimen on our microscope, the light is also going to bend. And so when you use the oil objective lens that I was talking about earlier, we must add a drop of oil. If you don't add a drop of oil when you are using the oil immersion lens, the light is going to bend. And why is that bad when the light bends? When you're using a microscope, the image is going to be very blurry. Okay, so let me show you right here to kind of explain to you what happens. So right here, here's the slide. So the slide is going to have the specimen on it. And down here you have your condenser and your iris diaphragm. Right here This is the objective lens, the oil immersion lens, okay, the 100x objective. Right here, let's say that this image is showing you what happens when you place oil versus when you do not place oil. Where do we place the oil, by the way? You place it on the slide. Okay, so you go ahead, you place your slide on the stage, and then with a dropper, you go ahead and you add a tiny drop of oil. Notice how when you place the drop of oil, the light doesn't bend. Why is that good? Because notice how when the light doesn't bend, it's going to hit the objective lens. Okay, and so actually that's going to make you see the image or your specimen very clearly. Watch what happens when you don't put oil. Okay, so do you see how here they're kind of telling you air, like you didn't put a drop of oil. Notice what happens to the light. The light bends, the light refracts. Okay, so the light refracts if you don't put that drop of oil, and so what happens is, notice how when the light refracts, it's not going to hit your oil objective, and so therefore the image is going to be very blurry. So make sure that whenever you use the oil immersion lens, you must put a drop of oil. Only, by the way, when you're using the oil objective. Okay. Right here, those are some other type of some other types of light microscope. So this one right here, it's we refer to it as the bright field microscope, and this one right here, it still uses visible light and everything as the source of illumination, and the specimen basically kind of like shines against a bright background. Okay, so against the bright background, and you are able to see the specimen pretty well. And this type of microscope can actually, show you a bunch of different types of, bacteria, for example. But it's not going to be used for viruses, by the way. Okay, viruses are too small. This type of microscope is not going to work for to view viruses. I'm sorry. There's another one called the dark field. And why do we call it dark field? Because you see how your specimen is going to shine against a very dark background. Okay, so it's going to appear against a pretty dark background. And dark field microscopes are also pretty good to examine living microorganisms that you cannot stain. Um, Microbes are clear, okay? They don't have a color. So for us to view, we have to stain the microbe. And so some microbes out there, though, they don't do too well when you stain them, okay? And so you would have to view them unstained. So this type of microscope would actually be ideal for those type of microbes that cannot be stained or don't get stained easily, or they get distorted when they get stained, okay? There's also the phase contrast microscope, and this one, it's pretty good to view details of the internal structures of your living specimens. You can kind of see actually their internal structures, which is very cool. This one right here, kind of the DIC microscope, it gives you three-dimensional images. Okay, so this one kind of gives you three-dimensional images of the specimen that you are viewing. Fluorescence microscopes, your specimens are actually going to fluoresce. They're going to not because they are fluorescent, okay, particularly, but you could actually have used a fluorescent dye, okay, so you've stained your specimen with a fluorescent dye, and so these microscopes will actually allow you to see, the fluorescence of these microorganisms after you've, for example, stained them with a fluorescent dye. Scanning acoustic microscope can actually show you living cells that are attached to surfaces. So, they use these type of microscopes, for example, when they're trying to identify cancer cells that are attached to your healthy cells, artery plaques, etc., biofilms, so bacteria that are sticking on surfaces, they can go ahead and use that. We have a bunch of different types of electron microscopes. So they use a beam of electrons instead of light, okay, to view the specimens. So they shoot those electrons, and that's how they're able then to see those specimens. This one is called the transmission electron microscope, and those are actually used to view viruses. So those are going to allow the visualization of viruses and also internal structures. Scanning electron microscope, they help you see the features that are outside the cell. So see right here how you can see, for example, like a bunch of cilia that are found, located on this specimen right here. So they're pretty good to help you study those surface features. Now we're going to introduce how do we prep a smear of bacteria so that we can go ahead and stain it. And we're going to discuss it more in lab, like we're going to go over those steps in more detail in the lab. But for now, I'm just going to go ahead and introduce you, okay, to the concept of how do we go ahead and prepare a bacteria smear. So that we can go ahead and stain it and visualize it, visualize our specimens under the microscope. So first of all, what's a smear, right? A smear is basically a thin film, okay? A thin film of microorganisms spread over a slide, okay? So if I want to create a bacteria smear, okay, then I'm going to take a sample of my bacteria and I'm going to go ahead and spread it on my slide. And you don't want too much, okay, of your specimen because the thinner it is, by the way, the thinner that film on your slide, the best, okay? If you put too much of your specimen on your slide, that's not a good thing. You're not going to see anything under the microscope. It's going to be way too much specimen, and you're not going to be able to at all figure out the features, okay, of the microbe that you are looking at. So it's best to have a very thin spread of that specimen on your slide, and we're gonna explain in lab how do you do it, okay, and what tools do you use. For example, this tool that you're seeing right here, it's called the inoculating loop. Okay, do I always have to use an inoculating loop when I'm creating my smear? No, you don't have to. Okay, there are other ways, different techniques. But the very first step, okay, of creating, a slide of a specimen is to go ahead and have a very thin smear or film of that microbe over your slide. Then it's pretty important that you leave it to dry, okay, because a lot of times those specimens, we're getting them from fluids, etc. It's pretty important that you go ahead and you let them air dry, okay? Even if they're not coming from fluid, we usually add a drop of water, and again, we'll explain, all of those techniques, but it's extremely important that you go ahead and you leave it first to air dry completely. So this process, do not rush it, okay? You leave your slide on, on your desktop, on your workbench, basically, and you leave it until it air dries, okay? Do not fan it, okay? Because that can actually aerosolize the bacteria in the air, and then, that could be dangerous because then you can inhale it. Okay, so just leave it to air dry. Now this step right here, we call it fixation. You're gonna fix your microbes. What does that mean? There's nothing broken, okay, with your microbes. What does it mean I'm gonna fix my microbes? That means that you're going to attach them to your slide. Okay, that's the term we use, fix. So fix meaning you're gonna attach your specimen to the slide. How do we do that? How do I attach my specimen to the slide? You take your slide and you just pass it over a flame 2 to 3 times, very quickly, by the way. Okay, so very, very quickly, you go ahead and you pass it over the flame so that it can attach to the slide. And at the same time, what's happening is the microorganisms are going to be killed along the way when you do that. Now, why don't you want to, you know, leave it on top of the flame for such a long time? Because that can actually aerosolize the bacteria in the air. And again, that's dangerous because then you can end up inhaling it. That's why it's super important that you do this step very quickly, okay? After you go ahead and you heat fix, and by the way, are there certain staining procedures where you must skip this step? Yes. Okay, so there are certain procedures where you should not heat fix, and we'll explain later, okay? And I'll tell you when you shouldn't heat fix, okay? And then after usually like, we heat fix, we go ahead and we stain it, okay? And as I said, sometimes you go from air drying directly to staining, okay? We'll talk about those procedures where you should skip this part right here. You usually flood your whole entire slide with the stain. We leave it also for a few minutes. Each procedure is different, okay? So some procedures are going to tell you you have to stain for a minute. Some procedures are going to tell you, well, this step you have to rinse it right away after 30 seconds. So, that's going to be discussed more in lab, okay? And then you usually rinse it, okay, to remove the excess dye. So you just rinse with some water and then you blot it dry very gently, okay? You don't want to dry it, too harshly, because if you do, you're just gonna take off your bacteria, okay, that you just spent so much time prepping. So you have to be very gentle when you're blotting dry your slide. And then last step, of course, is to go ahead and view it under the microscope. And as I said, with microbiology, we usually gotta go all the way to 100x to be able to see those bacteria cells. Now I want you to know or introduce you to positive staining versus negative staining. What's a positive stain? A positive stain is a type of stain where the dye that we use, it's usually known as a basic dye. What are basic dyes? Basic dyes are dyes that have a charge to them, and those basic dyes, the charge that they are carrying are positive. Okay, so positive stains are stains where we go ahead and we use a dye that has a positive charge to it. So let me kind of explain something to you guys. So let's say this is a bacteria cell right here. Now it turns out that bacteria, okay, most bacteria, their surface is negatively charged. Okay, so right here I'm going to put like a bunch of negative charges. So bacteria, their surfaces, okay, they are negatively charged. Okay, now if I want to stain the inside of my bacteria, basically like I want to stain that negative, charged bacteria. Now, if I use a stain that's positive, right, because this is a positive stain right here, what's going to happen? Opposites attract. Okay, so opposites attract. So if I use a positive stain, what's going to end up happening is this bacteria, okay, right here, let's say I'm using a blue dye, this bacteria is going to take in that stain and it's going to be stained blue. Okay, why was the blue stain attracted to that bacteria? Because the stain is positive, the bacteria is negative, opposites attract. And this is what you're seeing right here. See how you have a bunch of circular bacteria, and we use the term cocci, right? Or coccus, singular, cocci are plural. So you see a bunch of cocci bacteria right here, and notice how the bacteria was the one that got stained, not the background. See how the background didn't get stained? It's the bacteria that got stained. And why was the bacteria the one absorbing that stain? Because the bacteria is negative and the stain that I used was positive. Okay, and so it got attracted to it. Now let's see this example, a negative stain. What's a negative stain? So let's say one more time, here is your bacteria, and again, it has a bunch of negative charges surrounding it. Now, if I use a stain that's negatively charged, okay, what's going to happen when you have two things that are negatively charged? They're going to what? Are they going to attract each other or are they going to repel each other? Well, they're going to repel each other. And so what's going to end up happening is the bacteria is going to, it's not going to accept the stain. Why is it not going to accept that stain? Because it's repelling it, because it's negative, the bacteria is negative. And so what's going to end up happening is the bacteria is not going to get stained, but what's going to get stained is the background. Okay, so the background around the bacteria is the one that's going to accept the stain, while the bacteria itself, it's not going to accept the stain. Okay, and this is what you're seeing in this image right here. Notice how the background is kind of dark. Do you see how here the background was not dark at all? Okay, versus here. You see how dark the background is? Because it's the background that got stained. And notice how the bacteria, it's kind of shining and illuminating. That's because there's a huge contrast, right? And because the bacteria, it's not stained, it's going to appear like, it's going to appear like fluorescing. Okay, even though we didn't use any fluorescent dye. But it's going to kind of give you an impression that it's like fluorescing against that, dark background. Okay, so this is called negative stains. What's a negative stain? A negative stain is when you go ahead and stain the background, not the bacteria itself. And the dyes that are usually used for negative stains are acidic dyes. Those are the ones that are negatively charged. So negative stains stain the background. Positive stain, they stain the bacteria cell itself. Okay. Now, there is a simple stain versus differential stain. What's the difference? A simple stain, we usually use a single dye, okay, and it's usually a basic dye. Remember, basic dyes are the positive charged dyes. So what are you staining? Well, you're staining the cell itself. Okay, so right here, you see how, one more time, you can see the cell itself got stained and you have a bunch of cocci. This image right here, just kind of showing you a different shape of the bacteria that was also, stained. And you can see that those bacteria, they look like rods. Rods, by the way, we refer to them as bacillus. Okay, so those are bacillus in shape, meaning rods. And what's a simple stain used for? A simple stain is used to highlight the microorganism so that you can visualize it. So you can tell what shape it is. I mean, right here, I'm able to see it and I can see that the shape, it's a coccus, it's circle. Again, right here, I can see it and I can see that it's bacillus in shape. So it can help you figure out the shape, the structure, the arrangement. And we're going to talk about arrangements, are they clumped together, staff flow, or, we're going to talk about the different names for the arrangements, but this is the whole point of a simple stain, so that you can go ahead and basically take a look at the microorganism. Okay, now what's a differential stain? Differential stain is actually going to help you distinguish between different types of bacteria based on certain things that we're going to talk about. So some differential stains that we have are gram stain, acid fast, and spore stain. So let's take a look at each. A gram stain. What is it going to help me distinguish? How is it going to help me distinguish, the differences between bacteria? It's going to help you distinguish the bacteria that has a thick peptidoglycan cell wall versus the bacteria that has a thin peptidoglycan cell wall. The ones that have a thin layer of peptidoglycan, we are going to refer to them as gram-negative. The ones that have a thick peptidoglycan, those are going to, we're going to refer to them as gram-positive. So this is an image to kind of show you what I'm talking about. So this is the plasma membrane. Okay, so both gram-positive and gram-negative, they do have a plasma membrane. And then they have a cell wall. Now, this is the layer that we are talking about, the peptidoglycan layer in the cell wall. Notice how some bacteria have a very thin layer of that peptidoglycan. It's a very thin layer. Okay, they have some other stuff, but in terms of the peptidoglycan, they have a pretty thin layer of peptidoglycan. And then this one right here, this bacteria right here, notice how thick its peptidoglycan is. The ones that have a thick layer of peptidoglycan, we refer to them as gram-positive. The ones that have a thin peptidoglycan, we refer to them as gram-negative. Okay, we are going to talk about the gram staining procedure in more detail in the lab. But again, I'm just going to introduce you, okay, to what the gram stain is, quickly kind of go over some of the steps, okay, or all the steps of the gram stain, but and not, briefly, okay. Let me show you. Okay, so the first step is you go ahead and you put your smear. Okay, remember the smear got to be pretty thin on your slide. Okay, and then let's say you have two slides. Okay, and on one slide you put a thin smear of bacteria. On another slide you put another thin smear of bacteria. After you go ahead and you place your smear and you air dried it, etc., and you heat fixed it, by the way, if you take a look at the bacteria under the microscope after this fixation step, they're both going to look clear, okay, because you haven't stained them yet. So the first stain that you're going to use is, we refer to it as the primary dye, okay, because it's the first, so it's the primary dye. And we usually use crystal violet, okay. Crystal violet, it's a basic dye because, as you can see, it's going to be accepted by the bacteria. So the bacteria is going to accept it, and so the bacteria are going to look blue, or kind of like, bluish, purplish kind of color under the microscope. Okay, like a violet color, basically. Okay, and then what we do is you go ahead, you rinse the crystal violet, and then you add some Lugol, okay? Lugol, by the way, it's iodine, okay? So Lugol and iodine, they're the same thing. You go ahead and you add iodine. Now, iodine, we refer to it as this term right here that you guys see, mordant. What's a mordant? A mordant is, it could be a chemical, or it could be some other sort of technique other than adding chemicals. It could be heat, and I'm going to explain to you how a mordant could be heat. But in this case, what's our mordant? In this case, our mordant is iodine. What's a mordant? A mordant is a step where it's going to help that primary dye get absorbed better by the bacteria, okay? And it's going to make it tougher for that stain to get rinsed off. So what's the stain that we really want the bacteria to absorb very well and not get rinsed pretty easily? The crystal violet, right? That's why I added the Lugol after adding the crystal violet. I'm trying to, allow my bacteria to take in that crystal violet and not lose it after, I rinse. So after the iodine, you leave it for a specific amount of time. You go ahead and you rinse it, okay? What do we rinse it with? We rinse it, you can rinse first with like some water, and then you can go ahead. This is a pretty important step. You must add alcohol. So alcohol or ethanol, this is going to be your decolorizing step, okay? Now, why is ethanol called our decolorizer? Because when you add your ethanol, notice how those that have a pretty thin layer of peptidoglycan, they're going to lose the crystal violet. You're like, but wait a second, I thought the iodine is supposed to make it hard for that crystal violet to be washed off. Correct. But it's only going to make it hard, to be washed off if the bacteria has a thick layer of peptidoglycan. If the bacteria has a thin layer of peptidoglycan, it's going to be washed off. Okay. Now, so after that decolorizing step, this gram-positive bacteria, it's going to still look purple, okay, while the gram-negative, because now they lost the crystal violet, they're going to be clear. And so how am I going to see them, right? You have to go ahead and use a counter stain. So the counter stain, it's going to be safranin, and safranin is reddish in color. Now, so you add the safranin, okay, you flood your slide with safranin. The gram-negative, they're going to accept it because they're clear, right? They're going to accept it and they're going to look red, while the gram-positive, they're not going to take it. Why? They're already saturated, okay? They're already saturated with the crystal violet. And so this is how you can differentiate between bacteria that have a thick layer of peptidoglycan. They're going to appear violet under the microscope. And those that have a thin layer of peptidoglycan, they're going to appear red under the microscope. Okay. This is a table kind of showing you, the steps of the gram stain one more time. So one more time, you start off with clear cells. You add your primary color, the crystal violet. They're both going to accept it. Both cells that have a thick layer or a thin layer of peptidoglycans, they're both going to be purple. And then you add the iodine, right? The mordant, that's going to help your crystal violet, get absorbed even more and kind of make it harder for that crystal violet to be washed off. So after that step, they're still looking both purple. And then you add your decolorizer, okay? And your decolorizer, you got to be very careful how long you keep that decolorizer on. We're going to talk more about that in lab. So you add your decolorizer, which is your alcohol, okay? And that decolorizer, okay, ethanol, it's actually going to allow the crystal violet to actually be washed off, okay? Only from the cells that have a thin layer of peptidoglycan. The ones that have a thick layer of peptidoglycan, they're not going to be affected by that decolorizer. But it's really important that you don't keep the decolorizer on your slide longer than intended. Because if you keep it on for, you know, right here. You're supposed to only keep it for 20 seconds. Let's say you placed it on your slide for 2 full minutes. Guess what? Even the thick layer that has the crystal violet is also going to be decolorized. So that's why it's extremely important that you don't keep the decolorizer on for such a very, you know, for such a long time. And then the last step is to go ahead and add your counter stain. Who's going to accept the counter stain? The cells that have a thin layer of peptidoglycan. So they're going to accept the stain and they're going to appear red under the microscope. So this is an example right here. The ones that are appearing red, those are gram negatives. The ones that are appearing, you know, violet, those are the gram positive. Okay. Now there's another type of differential stain, and that's known as acid fast. So acid fast, it's going to help you differentiate between bacteria that have a waxy layer in their cell wall. Okay, so notice how here we're not talking about peptidoglycan. Okay, we're talking about that waxy layer. So it turns out some bacteria have a waxy layer in their cell wall. There are actually two genera, like you know, two genus, okay, genre of bacteria that have that waxy layer in their cell wall: Mycobacterium and Nocardia. Okay, an example of a Mycobacterium, you know, bacteria, Mycobacterium tuberculosis, the bacteria that causes TB, does have the waxy layer. So let's go ahead and take a look at the acid fast stain procedure. So the acid fast stain procedure, again, one more time, you got to start with a smear. Make sure your smear is thin on that slide. You're going to go ahead and add a primary stain called carbylfuchsin. Okay, carbylfuchsin is red in color. Okay. All cells at this point are going to appear red. Okay, so whether it's an acid fast or a non-acid fast, they're both going to appear red. You add a decolorizer. Okay, actually before the decolorizer, what's your mordant? Is your mordant iodine? No. Your mordant in this case, it's heat. So the mordant can actually be heat. And remember again, what's the mordant? The mordant's job is to help that primary dye get absorbed by your cells even more, and kind of, you know, makes it harder to get washed off. So we add heat, like you take your slide and you actually place it on top of the flame. And why heat is used? Because you're trying to penetrate that tough waxy layer. So that tough waxy layer, you know, heat would be a pretty good, you know, way to penetrate that tough waxy layer, so that the so that the carbylfuchsin can be absorbed. Okay, better. And then you go ahead and you decolorize with acid alcohol, not ethanol. It's acid alcohol. By the way, with each procedure, you got to make sure that you are using the correct decolorizer, because if you're not using the correct decolorizer, things are going to go wrong. Okay. Now, the ones that do have the waxy layer, they're not going to be decolorized. They're going to maintain that red color. Those that don't have the waxy layer, they're going to lose the carbylfuchsin. Okay. Once they lose it, they're going to become colorless. So what do I have to do to view them? I have to add a counter stain, such as methylene blue. And so the ones that are going to accept that blue color are the non-acid fast, which are the ones that don't have the waxy layer. Okay, the ones that do have the waxy layer, they're going to appear red under the microscope. Those that don't have the waxy layer or we refer to them as non-acid fast, they're going to appear blue. So this is an example right here. The ones that are appearing red in color, those are the ones that have the waxy layer. Those are called acid-fast bacteria. And this is an example, Mycobacterium bovis. Or as I said, the Mycobacterium tuberculosis is another example, okay, that have those waxy layers. The ones that appear blue, those are the ones that we refer to as non-acid fast. So do you see how this is a differential stain? And again, it's differential because I'm able to differentiate between the bacteria that have the waxy layer versus the ones that don't have the waxy layer. That's why it's called a differential stain. There are some stains that we refer to as special stains because they help us distinguish specific parts and they help you view specific parts within the microorganism. So we have capsule stain, endospore, or you can refer to it as endospore or spore, okay, stain. And then we have the flagella stain. So capsules, what are capsules? We're going to discuss them more later on, but basically capsules are a covering that some bacteria have, okay, around them. And those capsules actually give advantage for bacteria that have them. They can actually withstand harsher conditions and it kind of helps them survive, okay? Harsh conditions, low nutrient levels, etc. And having a capsule, usually it's also going to therefore increase the virulence of the bacteria. So usually bacteria that have capsules, they're going to be pathogenic. They're going to be disease-causing bacteria because the capsule is also going to help them evade your immune system. How do you see a capsule under the microscope? You gotta do a negative stain. Remember the negative stain where you use acidic dyes to go ahead and stain the background? So you stain the background, and at the same time also you do a positive stain and stain the bacteria itself. But because the capsule, it's a pretty tough, hard shell, it's not going to accept any of the stains, okay? And so what ends up happening is you end up with this halo look. Do you see how here is my bacteria, that kind of purple one, and then there is this like white halo around it? That halo is the capsule. Okay, so if there was no halo, that would tell you the bacteria doesn't have a capsule. But if you do see that halo after doing, the capsule stain procedure, and you end up with that halo, that tells you that it has a capsule. Endospores are basically resistant dormant structures that are formed within the cell, within a bacterial cell. And those endospores, they basically protect the bacteria from harsh environmental conditions, especially when the bacteria realizes that, let's say, it doesn't have enough nutrients, etc. The bacteria is going to start producing those endospores. Usually endospores are produced by older bacteria. So the older the bacteria gets, okay, the more endospores are going to be produced. So these endospores, they're also resistant to a whole lot of stains, and so they don't get stained also easily, okay? So how can I go ahead and stain those endospores? So let's say right here you have two types of bacteria. This one right here is producing those endospores inside of it, so it's like endospore positive. This one it's not. So again, they're both lacking color. So you go ahead and you add your primary stain. For example, we go with malachite green. So they're both going to get stained green, okay, including the endospores. So you see how like the whole thing gets stained green. And then you go ahead and you decolorize with water. Okay, so you go ahead and decolorize with water. And what ends up happening is when you decolorize with water, the cell, the bacteria itself, is going to lose the malachite, but not the endospore. The endospore, it's pretty tough. Okay, so after that stain has gone into the endospore, it's not going to be an easy task to actually take it out of the endospore. So it's going to stay inside. Okay, so then what happens is we want to go ahead and stain the cell itself. So we add a counter stain, and that's going to be safranin. And safranin is red in color. So what ends up happening is the bacteria is going to be red in color, and then the endospore is going to stay green. This one right here, because it doesn't have an endospore, the whole thing is just going to look red. So right here, you can kind of see, let's take a look at this image right here. Now, do you see how there are right here bacilli, again bacilli meaning rods, bacilli shaped bacteria that don't have spores, and then right here, do you see how some of them have like green inside of them? And that green inside of them, those are basically the spores. Okay, so that's basically a special stain right here, and you can see the spores inside of the bacteria. And by the way, these spores eventually are going to be released out. So if you see like also the spores on their own, not inside of the bacteria, that's absolutely normal as well. The last stain is the Flagella stain, and that stain is used to highlight the flagella of the microorganism that you're viewing under the microscope. So if you want to go ahead and view those flagella, you also gotta stain them. And flagella is basically structures of locomotion so that the microorganism can move.